Today we're diving into a hidden data trap that can literally flip scientific results on their head. It's this really sneaky statistical error that lurks in all sorts of research, causing scientists to see something decreasing when in reality it's actually increasing.
So get this. In a huge meta-analysis of multiple cancer studies, the standard way of doing things showed a particular molecule decreasing inside tumors. But when the data was looked at in the right way, the truth was the complete opposite. That same molecule was actually increasing, and not just by a little bit.
So how on earth can the same data tell two completely opposite stories? Now, this isn't some mistake in the lab work, right? The measurements were fine. It's all about how we read the data, and we're about to crack this case right now.
And the entire mystery? Well, it all boils down to one simple but absolutely critical difference. Are we looking at the actual absolute numbers of something, or are we looking at them as a percentage of a bigger whole? Because that distinction, as you're about to see, changes everything.
Okay, let's make this super simple. Imagine we have a fruit basket. If we talk in absolute terms, it's easy. We count them up, we've got 5 apples and we've got 5 oranges. The actual number of each is 5. Simple enough, right? But what if we look at this in relative terms, percentages? Well, there are 10 fruits in total. So the apples make up 50% of the basket, and the oranges make up the other 50%. Nice and balanced, 50-50.
Okay, now here's where the magic trick happens. We're going to add 10 more apples to our basket. But listen, we're not touching the oranges, not a single one. The number of oranges is still 5. We just have more apples now.
And boom, this is the trap. Look what just happened. The apples now make up 75% of the basket, and the oranges, even though we still have the exact same 5 oranges we started with, their percentages suddenly dropped to just 25%. From a relative point of view, it looks like they're disappearing.
And that's the whole point. If you were only looking at the percentages, you'd be screaming, the oranges are decreasing. But nothing biological happened to them at all. It's just a quirk of the math, a mathematical necessity. When one part of a fixed total goes up, the percentages of everything else have to go down.
Well, in the world of data science, this kind of parts of a whole data has a name. It's called compositional data. Anytime your data adds up to a meaningful total, like percentages or parts per million or market share, you're dealing with compositional data.
And this is where things get really dicey for researchers. Most of the statistical tools we use every day, they weren't built to handle this stuff. Using them on compositional data can lead to false positive rates of over 30%. So you get scientists chasing these ghosts in the data, finding significant changes that are just illusions, just like our disappearing oranges.
Okay, so if the usual tools are leading us astray, how do we fix it? How do we get out of this trap and actually see what's really going on in the data?
Well, the answer is a really clever approach called compositional data analysis, or CODA for short. Instead of getting fooled by the percentages, CODA looks at the ratios between the different parts. It lets you see how things are changing relative to each other, which sidesteps the whole fruit basket problem entirely.
And the way CODA works is actually pretty logical when you break it down. First, you just have to recognize you're working with compositional data. Then you use some special math to focus on those ratios between the components. You also have to account for any changes in the total amount, the overall size of your basket. Once you do that, you can run your stats and get a much truer picture.
All right, so now that we have our secret weapon, CODA, let's go back to that cancer study that started this whole thing and see what happens when we use the right tool for the job.
And just look at this. It's a tale of two completely different stories. On the left, the standard analysis shows a decrease, but on the right, the CODA analysis reveals the truth, a huge increase. The conclusion isn't just different, it's the exact opposite.
And this isn't just about being right or wrong. It has huge implications. When they used this molecule to try and predict cancer, the old method gave it a moderate accuracy score of 0.69. But with CODA, that score shot up to 0.81. That's the difference between a pretty good predictor and a really strong one.
Now, I know what you might be thinking. Okay, that's a cool story about one cancer study, but how often does this really happen? Well, the scary thing is, this trap is hiding in plain sight everywhere.
Just think about it. Microbiome studies, looking at the percentage of different bacteria in our gut, gene sequencing, analyzing relative gene expression. Heck, even economics, when you're looking at the market share of different companies. All of these fields are swimming in compositional data, and all are at risk of this exact same mistake.
You know, one of the most interesting things from the research paper is this wild paradox.
For years, the fact that many studies were kind of small actually protected them from this error. With a small sample, the false findings weren't powerful enough to look statistically significant. So in a weird way, smaller budgets actually saved them from making a ton of mistakes. They got lucky.
But here's the kicker. That luck is starting to run out. As we enter the era of big data and our data sets get massive, this problem actually gets worse, not better. It creates even more convincing false alarms. So the real question is, as we collect more and more data, how many other traps like this are just waiting to be discovered?